Let me take you back to 1940, where these three researchers came up with an experiment that would actually be a big contribution to the systematics, ecology, and the genetic world. Three men, Jens Clausen, David D. Keck, William M. Heasey, decided to study a large number of plants from many unlike species and families. Since there are multiple species, they come from a wide range of habitats, ranging from these forms: maritime, coast range, foothill, mid-altitude, subalpine, alpine, Great Basin, and plants from high latitudes. Now, I'll give the mic to them. For your information, we had to observe these plants for up to 20 years in 3 of our selected gardens: Stanford, at the university that me and my boys worked at, Matter in Sacramento County, which is about 4,600 feet above sea level, and Timberline in the Sierra Nevada, which is about 10,000 feet. As this study is very broad, we picked out a few important results from our studies, and we'll focus on the subalpine and alpine forms. They differ in their height, since alpines are usually dwarfer, and they flower earlier and are more often frost-resistant than subalpines. When these forms are transplanted, we observed that in Stanford the time of flowering was delayed, but this was not the case in Matter. Another question is, do they survive in any of these gardens? All subalpines and most alpines increase in height and vigor at the mid-altitude stations, but lessened in Stanford. They survive at Timberline, but only alpine seed matures there. This might correlate directly with their frost resistance. What they found out is that these transplant experiments have shown that regional races are not environmental modifications, but are heritably different. The second important study that we conducted was observing how chromosome number for various species have any role in their character differences. When there are no differences in the chromosome number, we found out that 2 different ecotypes, morphological and physiological characters, showed genetic segregation. We will look at the hybrids between Zooxantheria. There is a differentiation that is produced by numerous genes of the multiple type. Their characters evidently depend on a series of genes with minor effect, and the existence of genetic linkage between taxonomic characters is shown below, and those of importance for survival were highlighted. We can also point out that absorption of genes from related ecospecies may contribute to the variation. When there are differences in chromosome number, free intercrossing between the group is prevented, and a species differentiation has been set up. Here we have the xenospecies Viola purpurea, and they contain 2 ecospecies with different ploidy: a tetraploid in the coast ranges and Sierra and foothills, and a diploid in the Sierras and the Great Basin. The latter develop morphologically distinguishable mid-altitude, subalpine, alpine, and Great Basin forms. Hence, this diploid ecospecies shows the greatest variation and ecologic adaptability. However, potentials of xenospecies Drummondii and gracilis differ due to chromosomal irregularity with high polyploidy. They vary in chromosome number within the ecotype and even with the population. Nevertheless, despite the oscillating chromosome number, the ecotypes are reasonably differentiated. From this observation, closely related plants differing in chromosome number usually occupy different environmental niches. This was also studied by another researcher 70 years later, who did a research on Claytonia parviflora, and obtained results that suggest that polyploids occupy distinct and broader niches relative to diploid, but of course this would vary within different species. Our work have a huge importance in the study of epigenetics. Epigenetics is the study of how an organism's behavior and environment can cause changes that affect the way their genes work. Results are shown due to transplanting of flowers in different environments, which gave various observable outcomes. The outcome of individual plants does not modify or give permanence, but have shown to be reversible. This is important in the study of epigenetics and stresses on how environment and behavior changes does not change our DNA sequences. Clausen used this finding to do another research to back this up that focuses on Potentilla glandulosa. As this work is filled with a lot of broad findings, it would be a great base for evolutionists to discuss any aspects of specifications in which they're particularly interested in. Can you see how this work plays a huge role in genetics? So can I. Thank you.